Hello, everyone. We are once again in the What We Are Reading YouTube channel for the South American Mycorrhizal Research Network. I am Ana Bermudez. I am a PhD student from the Karst Lab at the University of Alberta. And today we have Dr. Ariadne Furtado from the Federal University of Paraíba, who will talk with us about her recent paper that is titled Guapirioid ectomycorrhiza, a novel fungus-plant subtype is described associated to Guapira opposita, Nyctaginaceae in the Brazilian restinga. Welcome, Ariadne.
Hi, Anna. Thank you very much for having me today. For me, it's really important to be here. I'm really happy. I value very much this kind of initiative by the South American Mycorrhizal Network, which is being very well led by Dr. Cesar Marin and has the support of several researchers, such as Dr. Camille Tragon. And I mean, I just wanted to say that I believe that this interview are really important because they kind of bring us closer, especially to those that are super curious about what has been studied about mycorrhiza around the world. And the important thing here is for them to see that we are people behind the surnames and manuscript, and even more important for those that are just starting out, for instance, the undergrad students. So yeah, thank you very much for this, for having me for this, to talk about a little bit more about my research.
Thank you for being here. And yeah, I'm excited to ask you some questions. Talking about why do you think it's important to characterize the fungal diversity and the mycorrhizal associations in the neotropics, but in particular in this region where you worked in the Brazilian restinga?
From the beginning, I'm a mycologist. I am major in biological science and I studied mushrooms. It's been 15 years now. And I started, as several of us, with taxonomy. And I understand that to understand how communities come together and how the organism and their multiple strategy to resist environmental conditions, how interspecific interactions add another layer of complexity to the structures of this community. How all of this is happening, you need to know who is participating of these communities, who are the animals, plants, microorganisms in general, fungi, etc. So the first steps is to recognize these organisms. So especially where their conditions are limited, the cycling process, for instance, like the restinga, which is a really particular environment, extreme environment, they require specific characteristics to be maintained by all the organisms that are there. So the restinga was one of the first environments to be harmed by human intervention in Brazil. And currently we have less than 80% of the Brazilian coast covered by the restinga. And the restinga is part of the Atlantic forest. And when you talk about the terms of biodiversity conservation status, we know nothing about it in the restinga, you know. And it's not only a really beautiful ecosystem. When you go into the restinga, you see how different it is from the Atlantic rainforest or the Amazon. You will see ecosystem with a sand soil, acidic, with nutrient poor soil that often has a high salinity because it's It's really close by the sea.
Mm,
It was like a buffer between the ocean and the forest.
mm.
So the plants there, they like manage to establish and survive in this sort of ecosystem. And the fungi, the ectomycorrhiza fungi, may function as a key component that allows it. And it also stores rainwater, controls floatings, regulate the water cycle, etc., etc. So you see that just starting by identify the fungal diversity and the mycorrhizal diversity, you're not just doing that. You like allowing yourself to know a whole ecosystem, and all this, recognizing all this importance.
Right. In this area, the restinga, did you have like a reason why you chose to work with that particular species of plant that formed this association, which I think is Guapira opposita?
Well, as we were talking, like plans change during our PhD.
Yeah.
The main idea was to investigate ectomycorrhiza interactions in the restinga, because it is, well, a really threatened environment. And I have worked previously in restinga. I've collected several different kinds of mushrooms, ectomycorrhiza mushroom, but we were mostly focused on the sporomes, not in the association per se. We were saying, this is ectomycorrhiza because, I mean, it's part of a family, a lineage that has ectomycorrhiza fungi. But we were not proving the association. This was not the plant, was the fungi.
Okay.
Because I'm a mycologist. And it changed because I was collecting at least 2 times per month. And once I started to work with the molecular identifications, most of the results from the plant side was Guapira opposita. And it was not because I was after Guapira opposita, because the results were coming for, it was Guapira opposita. And we do have a few ideas why this is happening. It's because one of them is because Guapira opposita, it's a Nyctaginaceae. It was already proved that this is a plant that works as a nurse plant. It acts like facilitating other plants to establish in the restinga. So in this area, you will see Guapira opposita forming a sort of a patch. And ecologically, we even might say that ecologically, the species is working as a monodominant species. You see, you will have other families, possible potential ectomycorrhiza over there, like Fabaceae, Phylmyrtaceae, Polygonaceae. But most of the results were fungi associated with Guapira opposita. And I was not collecting only like, Guapira opposita individual, I'm going to collect. No, I was doing like more random collections. And again, I was focused on the mushroom. So I was basically, you have this sporome, I was collecting under the sporome. So this is the results that I was accessing, was basically. So that's why we decided to focus in Guapira opposita. And also because the study the group of research that we have in South Brazil, we also have a physiologist, and he is particularly interested in how ectomycorrhizas help Guapira in his role, in its role as a nurse plant. So we are using Guapira opposita as a model as well, but at the beginning it wasn't the idea.
Okay, so it was more thanks to your work that you were able to identify it, no, as a important. Yeah. Okay. So now I think we have the basis of the system more or less understood where of this system where you work. So now I would like to go into more of the mycology and taxonomy aspects of. So for me, I was really impressed with all of the details and the descriptions and the pictures and all of the these aspects that are required in a study like this one that you did, no, that not only looks at the morphology and the anatomy of the association, but also I think you identified you also did molecular and phylogenetic analysis, no, of these fungi. And so I just wanted for you to talk a bit about all this process and perhaps what were some of the challenges, because I bet there were, it seems like a lot of work. So yeah, of all of these steps.
Yes, indeed, it was a lot of work. And this is why these moments are like actually really interested, like this kind of talk, because once you see the paper, it's pretty rare when someone said, oh, I struggle with this. Most of the time it's just perfect over there. And no, I struggled a lot in several things, because I mean, in Brazil right now, is studying ectomycorrhiza, the focus of the association. There's basically our research group in South Brazil. I was the first PhD student to perform like a bigger project, to develop a bigger project on the diversity of ectomycorrhiza fungi. So as the first one, I have to adopt a lot of methodology. So I have a lot of problems from the first step, from the sampling to the last one, which is the molecular analysis. The bigger challenge was to go after methodologies in literature, and everything that you find there is basically for you to access material sample collect in temperate ecosystems.
Okay, yeah.
And once you come for a different kind of ecosystem, which is multidiverse, the soil is completely different, you cannot apply that methodology. So you have to adapt to change several things so you can collect the more and preserve the more samples you can. So this is what the first thing that I have to change the methodology. I was going after papers and everything. I was changing everything. This doesn't work for me. This doesn't work for me. So I basically was using my own methodology by the end. And the second one was to do the sort of the how to work with the samples, because when you go after, I don't know, ectomycorrhiza systems in the Google images, you will see a beautiful branching thing, small one, amazing.
Yeah.
So when you're looking after this in the samples we've collected here in neotropical ecosystem, you won't see that. You will see not that branched system, really thin. Sometimes the mantle is barely imperceptible, really inconspicuous. This is the second challenge, to train your eye to don't discard everything. You have to check, double check and everything, because it will be different. Don't go after the ectomycorrhiza system thinking about the ones that you found in Ager's book.
Yeah,
this is wrong. And so I've lost a few materials at the beginning because of that.
Yeah.
And for the molecular, so everyone has a different kind of methodology for the molecular of ectomycorrhiza all over the world, even neotropics. And I started trying to use the extraction kit to access the fungi DNA and plants DNA. But when I tried to access the fungal DNA, I was accessing the plant DNA instead. It wasn't going like as I expected. So I got connected with this Italian group, which has been incredibly supportive with us, including Andrea Rinaldi was my co-advisor during my PhD, because I mean he helped me a lot with this to adapt methodology. So we understand it was easier if I access the plant DNA doing a normal extraction step using HIT or subtype methodology. But to assess the fungi DNA, I will have to forget about all the first steps and go direct to the PCR. So I was taking the tip, making the scalp of the mantle. So I was leaving the root naked. I would take the mantle and put on the Eppendorf and then do the PCR from there directly.
Oh, okay.
Of course, I had to use, for instance, bovine albumin to improve the reaction and access the DNA. Of course, this is kind of tricky because you have to use the exactly amount, otherwise you're gonna fail completely. And that's why. So I struggled the first step, the second step, and the third step.
How or at what point? Because I think so this is the highlight, or one of the highlights of the paper, is that you were able to identify that it was a new subtype of ectomycorrhiza form, no? So when did you started to say, okay, this is a completely different thing, or what made you think that it was different from other types of this association?
Well, the first one that I saw this happen, it was the species Amanita vistidolutea, which is a Brazilian native species from the Atlantic forest, from the restinga. But I couldn't confirm with just one case. I mean, I've collected once, twice, and etc. And then I collect again with other species, Austroboletus festivus, and then again and again and again. And then, this is why the morphoanatomical analysis was so important, especially the one where you can analyze with the transmission microscopy, because it was possible for me to prove that everything that we were saying, it was true. Because I mean, because we were insecure. I mean,
sure.
For years, someone was saying ectomycorrhiza is this, a mantle, hotignet, and extra radical hyphae. Who do you think you are to just say, no, perhaps?
Yeah.
And so we were trying to get prepared. So we were trying to have much material as we need to prove that. And I was completely sure that it was a different kind of the ones that we found, for instance, in pinus trees, etc. When there was the combination of well-developed mantle and no hotignet and no interradical hyphae. And we are proposing as a sub ectomycorrhiza type, because I mean, all the boxes, the names of ecto, this is something that we are creating. The nature is not like that.
Yeah, yeah.
And we have a few like results that said that, well, the ectomycorrhiza association, the mycorrhiza association, or for sure, like improve Guapira postdoc fitness. For instance, there's a growth in the aerial biomass, but we don't know how this the exchange, the nutrient exchange is happening, but something's happening. So and we have to perhaps update the concept of the ectomycorrhiza. The mycorrhiza is just when you have exchange of nutrients. Mycorrhiza also can happen just for the plant protection to pathogens, or I mean, there's several functions. I mean, again, all the boxes, this is our creation.
Yeah.
So all the combinations of the well-developmental and no Nyctaginaceae and no intraradical hypha, but with the proof of a biomass, this is all we have to say. It is a new subtype. Of course, there are the external characteristics, but it's more related also with the fungi species. For instance, to this long and unbranched ectomycorrhiza system, or it can be shorter, for instance. It depends. I think this is more connected with the species than the presence of any fungus over there.
Okay. Yeah, so now I just wanted to ask you if you are still working in that region, or what projects do you have now that you're working on, or what ideas are now in your brain that you would like to talk about?
Well, that's a few things happening right now. As I told you at the beginning, I am in half of a postdoc, and I'm really excited about this project because it's also with ectomycorrhiza. It's not about ectomycorrhiza from restinga, but it will everything that I'm learning from this, it will help me a lot to discuss all of this, all these particularities from Guapirioid ectomycorrhiza. I'm studying right now the mycorrhiza induced the small secret proteins, which are the MISPs, they'll call. They are protein effectors that are acting during the ectomycorrhization process.
Okay.
So because while I was studying, this is one of the papers that actually we were working right now, while I was analyzing the anatomy of the root, I saw that these particularities of no Nyctaginaceae, etc., and others, for instance, in the absence of a root epiderm layer, and all the changes, for instance, the cortical layer acquired, for instance, it changed to have bigger cells and bigger vacuoles, etc. All the change the root go through, it's in decorrence of the process that we call root determinacy. It's basically when a meristem get exhausted, and once you get exhausted, all the cells in the root tips differentiate to form a mature meristem. And there's several things that can motivate this. For instance, the presence of fungi associated with the root. And I really wanted to know how all these MISPs are involved in this process, because the signaling process between the fungus and the plant during this early and late stage of colonization will alter the expression of several plant hormones, and it, for instance, it might lead to the root determinacy. And I mean, you have to investigate auxin, salicylic acid, ethylene, jasmonic acid, etc. And we know nothing about which pathway are affected by the fungus signaling molecules or how they contribute to the plant hormonal imbalance. So understand all of this will help us for sure to comprehend more and more the morphological, molecular, and physiological aspects of the symbiosis. And I mean, there's so many more things that, because I mean, as I said, I mean, it's been a little bit more than 1 year that I finished my PhD, and I still have so much thirst of knowledge and so many ideas. And I really think that I need to dive a little deeper and like get acquainted with a few aspects of the universe of mycorrhiza. For instance, everything related with soil ecology. I'm really curious about trace-based approaches. So I can, for instance, predict frameworks of mycorrhiza dispersal, community assemblies, biogeography, and etc. And of course, we have, I'm connected with this great lab, research groups in South Brazil, the tropical ectomycorrhiza. There's a lot of really interested people over there, hard workers. But yeah, I'm also waiting for the results of a few projects that I'm submitted for grant. And they are focused on basically on investigate the diversity of ectomycorrhiza association. And in this dry ecosystem that we have in Brazil, for instance, Cerrado and Caatinga, to try to fulfill a little bit more this huge gap we have in Brazil, because if you see the map, you will see that you have a few spots in the Amazon and our own research group in South Brazil, but that's gigantic gap in Middle Brazil. So we need to make some effort to understand what's happening in this ecosystem as well. So it's basically this.
That sounds very interesting, and I really look forward to knowing more about your next steps. Yeah, so I think I will have to stop for now, but thank you so much for all that you've shared here with everyone. It was really interesting for me, and I bet for everyone that listens to this interview as well.
Thank you, Anna. Thank you so much.
Thank you.